Thank you very much, Emily and Jeremy. And I think a long time back, me and Jeremy worked for beginning the studies at Harvard. We started with flu peptides when proimmune was not as big as what is today. And he developed for me hepatitis pentamers for surface antigen. We did a clinical trial. So I have a long collaboration with him, and we're still thinking to do something, and I'll mention. So my talk is a little bit different than the epitopes, and I'm a cellular immunologist. So I give you a little bit, make you come out of your comfort box outside and see if you could appreciate where I'm coming from. So I'll give you a little bit because I have a different perspective than, immunology is here at Harvard is a humongous area, and there are a lot of people who have different kinds of expertise. I came here from India almost 40 years ago, and I started with infectious disease in UCLA. Before Cipro was accepted by FDA, I worked on ciprofloxacin, and then I did my tumor immunology in skin cancer, where Tucson has a very high incidence, and then moved to Dartmouth, did a lot of reproductive immunology, mucosal immunology, looking at it, and then taught medical students and went to India and came here in 2000 to Harvard, primarily to do the immune monitoring. So started with multiple myeloma, which is a neoplastic, one of the B cell, terminal cell, which is called plasma cell cancer, which resides in the primarily in the bone marrow, and it also located multiple, means it located in multiple locations, and it has a clonal proliferation, and it produces a monoclonal antibody. We don't know what is against for. Have a kidney issues, and then predominantly I identified with bone lesions. And these are the proliferating plasma cells. And these multiple myeloma could be any type of antibody they are producing. So it could be predominantly IgG, but it could be any of them, but you rarely see IgD or IgE myeloma. So if you could look at it, IgG is very common, and A is next, and the rest of them are very small. And there is a condition, pre-malignant condition, which is predominant in the older population, which has no symptoms, but they do produce monoclonal antibody. That would be very interesting. When I started 20 years ago, wow, this is easy one, we will figure it out, because they don't have the disease symptoms, but they have the disease component, main component, which is antibody. Only 10% of them will ever get multiple myeloma. 90% of them will never see anything cancer related. But it's not that simple, and I will explain to you. So there are 33,000 new cases every year in the United States, and 12,000 of them will die, and it is a significant portion of the cancer population, and slightly more common in men than women. This is a disease of older people. So just to give you a quick criteria, MGUS, the pre-malignant lesion, is predominantly less than 3 grams of the monoclonal antibody, and the plasma cells are less than 10% in the bone marrow, whereas the active myeloma has much higher and also associate with these clinical symptoms. So as you can see, as I just said, you can have MGUS condition and don't feel anything, and all of a sudden, boom, in 1 year, you start producing more monoclonal antibody and have bone lesions, and you have multiple myeloma and cancer, and you have a chance to die with it. So we try to generate multiple myeloma patients' immunobiology data. How do we monitor? Yeah, you can give the therapy. The another drawback in multiple myeloma is no matter what kind of treatment you give, almost 100% of them will relapse. That's another provocative question. So first we look at it, what is called PDC, which is plasmacytoid dendritic cells, which are the predominant source of type 1 interferons, and they also produce IL-3 responsiveness. So as you can see, PDC will make myeloma cells proliferate very easily and also make it resistant to the drug therapy, and also we did it in patients. And then we look at it, MDSC, myeloid derived suppressor cells, and as you can see, the myeloid suppressor cells are much higher in myeloma compared to healthy, and they will also help myeloma cells grow, which we all know MDSC are very important in the microenvironment, tumor microenvironment, how they pro-tumor. And then I look at it, originally I thought this is a plasma cells, right? No matter where you produce plasma cells in the body, they have a predilection or affinity to go back to the bone marrow, and that's where they produce antibodies only in a given time. Usually it's around 5-6 months. They're programmed to die off. How come these guys survived and keep on producing monoclonal antibody? There must be a regulatory mechanism is in vacuum. So I look at regulatory cells, and they are lower, nothing astonishing. Their FOXP3 levels are lower. We look at it, IL-17 in 2005, that was a very boom. IL-17, Th17 cells, which are required, IL-6 and TGF-beta, which are the two important cytokines that are required for multiple myeloma to grow. So I thought, wow, so maybe I could find something very interesting, and maybe I can find a cure. That's not that simple either. So even though we show, Th17 cells are higher, both in blood and bone marrow, significantly higher, and they also help their colony formation and growth and all sorts of other things. And how they help is because myeloma cells do express receptors for the cytokine. Then we did the animal studies. I mean, I have done at least a dozen compounds. These are a bone marrow environment replicating the human disease, and this is one of the best of all the drug treatments and new compounds, small molecules. This is one of the drug, but none of the drug companies are interested because they're a little bit afraid cancer patients, cancer trials are not that easy. It's easy to get drug approved for psoriasis or rheumatoid arthritis. So there are a number of companies who makes antibodies. They all have drugs approved for the psoriasis and rheumatoid arthritis. They don't want to touch cancer, but now they're interested, so we're trying to start a couple of trials with that. And we looked at further some of the PD-1 molecule expression, and they're all high, and they help. And PD-L1, if that is related to the multiple myeloma and how they are in patient samples. The interesting study compared to the MGUS, why they don't have symptoms, a Yale group did showing a SOX2, anti-SOX2, which is an intracellular embryonic antigen, which is not supposed to express, and how that intracellular antigen was seen by the T cells and produce antibodies is very tricky. So if you have those antibodies, your risk is much, much lower. Once they lose that, your risk goes up. We try to do that, and we did a lot of overlapping peptides. We wanted to make tetramers, but it didn't somehow it didn't work. But they also went on showing the PD-1 expression on the myeloma cells have something to do with that. So there is a humoral response because this is a B-cell related cell cancer. We wanted to do is that is there a humoral immunity defect in multiple myeloma patients. So what we did was we vaccinated with hepatitis B. So we vaccinated 22 patients, and very seldom, most of them don't have the response. We couldn't measure, even after 3 doses, we couldn't measure antibody levels. And if you compare to that, to the normal, and even the patients who responded, very low levels. It's only 100 versus 5,000 or so after 3 doses. So that tells you there is a humoral immunity compartment is somewhat damaged or very inefficient. So we are doing 2 huge clinical trials, 1,500 patients with a combinational therapy. Without transplant is one study. Another one is a combinational study. So this one is with a transplant, where we did was RBD with auto transplant, or with RBD, this is Revlimid, Velcade, Dex, which are standard drugs for multiple myeloma combination. And then following is the lenalidomide maintenance, and then most of them do relapse at the end of the treatment. It's all prolonging the relapse. You know, how soon you get the response, how fast they relapse. So this was published a couple of years ago in New England Journal of Medicine, showing that transplant has a better combination of the RBD with the transplant has a better prognosis. So in these studies, what I wanted to do is see if there is a B cell compartment is damaged or improved, or could we predict. You know, there's a lot of B cell tumors, as you can see it. Almost all hematology is in this B cell development group, whereas multiple myeloma is located all the way at the end in plasma cells. So there are a number of B cells generally located various places. They are called B1 cells or B1A. They are naturally, you're born with it. They are located in the peritoneal cavity. There are B1B cells and MZ cells. These are important in against certain capsular bacterial infections, and most of the cells that needs to go through germinal centers will acquire the switching capacity and also acquire the repertoire through SHM, and those are important. And so what we did was in the clinical trial that I showed, we took at the diagnosis, the frozen PBMC, and analyzed the B cell compartment about these 6 subsets. Are they damaged or they're inefficient, which will give us an idea. So you can see B1A, B1B, B2 cells are the ones will have full switching capacity, can produce immunoglobulin. As you can see, looking at the retrospective, these are the all diagnostic samples. People who responded have a high B2 level to begin with before the treatment. Those are the ones who responded. People who have very low, B2 cells in the beginning, and they don't respond. So that is a good indication. At least we're working on that and doing another 150 samples, which I will show you end of the talk, what we're doing with. And the GC maturation is a very important component for humoral immunity. There are light zones and dark zone in the germinal centers where a very active autoimmune presentation, autopeptide, autoepitope presentation by FTC to the B cells, and then T cells will come and talk to them in the dark area where they are eliminated, who are responding to autoantigen, and will only left who are not responding. That's why cancer is a very hard, and it's a very fine line. If you push too much, they're going to get autoimmunity. You might cure the cancer, but you might die with something else. So it's not that simple. And immune system is also very complicated. It's not that simple. We could just take a CD8 cells and show the killing activity in vitro. That is well and good, or transgenic mice, but in the body, immune system is not by itself and doesn't sit in the one room. Immune system is a very complicated and very important system, even though in medicine there is no separate branch for immunology. Okay, so I do talk to the dean once in a while, telling him. So So immune system literally goes every corner of your human body. It goes to every organ and involved in the cancer. All the autoimmune diseases are the drawbacks of the immune system. And the microbiota, which is a very hot area right now, what you eat, what you have in your gut, will determine how well we can tell who is healthy, who is not healthy. We teach a medical student, we all have infections today, but none of us have diseases because we don't have a prodrome or we don't have symptoms. So that doesn't mean that immune system is constantly fighting. And the brain is another one. We all think that CD8 cells have all these suppressive markers. They also have neuropeptide receptors. They are very reactive to the female hormones, male hormones, and we're working with the PTSD patient to show that. And I will show you somewhere and see if I can wake you up. And of course, infectious disease has been shown, our association with the immune system. We don't appreciate is this compartment. Is we only need 2,000 calories. I'm sure we eat 3 or 4 times higher than that every single day. And we all need to mobile. It tells you, CDC and everybody else, you need to walk 10,000 steps. We hardly walk 1,000 steps. And no wonder we have Alzheimer's and Parkinson's, and we can't remember after 60 what's happening. So immune system will tell you. So, I run 10 miles every single day because that's what I believe in, what I do, I practice. So because of that, it's important. So immune system is complex. Don't take it granted that you could show it in a Petri plate or in a ELISA plate. I have done this 40 years. Okay, so I will show you before I show you why I want to study in detail. Okay, I have struggled last 20 years in this town to convince psychiatric people inflammation has some role in the behavioral psychology. So this paper came in 2 years ago. It tells you that social behavior has extremely controlled by the production of gamma interferon, which is the salient cytokine in the entire immune system. Okay, we did some studies or doing it. If you look at it, this is healthy patients. These are CD4 cells stimulated for 6 hours, looking at gamma. And these are the non-CD4. If you look at the PTSD, CD4 cells have a high gamma production, and non-CD4s have almost half. So it's not the gamma production. It's a gamma production by who? May have something to do with it. If we look at it, NK cells, KIR molecules are very important, whether NK cells will do the surveillance properly, efficiently. So if you look at it, this is one of the KIR molecules. It's very high in PTSD compared to normal. So we're looking at the detailed immune function. PTSD, why they have those things, how traumatic brain injury, whether you have a physical injury or not, how is the inflammation play a role and override those things. And this is related to the sleep. We have a mouse model with NLRP3 knockout mice, which are very efficient in the inflammatory model to show that. So if you take a control mice, there are no B cells. This is in the brain. And if you take the sleep-deprived mice, and why are there so many B cells generally don't go to the brain? So there is something to do with it, if you think about it. So it is not that simple. You can't just study one marker and show that PD-1 antibody, I can cure cancer. That's, for me, it's very ludicrous. Even though the guy who made that, Gordon Freeman here, still agrees for only 20% or 25% may have a response. So if you look at it, there are so many markers we don't even look at it. You only look at what you are interested or what is published in this study. We are looking at B cells, memory, are they switch from IgM? Most of the B cells that come out of the bone marrow will only have IgM. So are they switched to G or A? You need to know that information. Or are the transitional cells are sufficient enough to go to germinal centers to produce mature B cells, or the precursors are in place because multiple myeloma tumor cells sit in the bone marrow where B cells are developed. And there are a number of them. These markers tell you those B cells gone to the GC. These markers will tell you those gone to the dark area, and this marker will tell you they're going to die. So like that, in T cells, number of markers, and we do a lot of NK markers. There are innate lymphoid cells. There is a new monocyte set came in a month ago in Science, which tell you CX3CR1, which is very important to develop inflammation or not inflammation. And there are 6 different DCs, which we all say there are DCs and take for granted there's only one DC type. And even though in mouse we know that there is a DC10, which produce lots of IL-10. So we are characterizing and these. And I'm just doing it in multiple colors. So these are all the B cells. You could see them. So we take the CD19 population, put it on 43 and 5. And these are other subset, switch or non-switch and naive. And looking at the transitional cells, these are the TFH gone to the GCs. And looking at those are in GC. These are in the dark area. These are going to die. And looking at the T cell subpopulations, and these are all, we will do the CD4, CD8 memory populations, and looking at the, I'm only showing 6 of them. We have another dozen cytokines, so we can do it. And then looking at the activation markers, 41BB, and also there are a dozen suppressive markers on each of these. And if you're looking at the Tregs, we all characterize, these are the Tregs, there's only one there. No, they express dozen markers. It depends upon what they express, how they function, what cytokines do they produce. Do they produce TGF-beta or IL-10? Are they expressing in combination of PD-1 or TGIT or LAG3? Will depend upon how they function, how the homeostasis is created. If you don't know all that, there is a guy presented at Harvard Medical School a couple of months ago, said that he can tell by looking at Tregs whether you heal in the bone fracture or not. I asked him, how do you know which Treg will be healing or not healing? He said, sorry, I didn't look at those. So it's not that simple. So looking at the TFH, even those cells that talk to the B cells in the germinal centers have several subpopulations. IL-21 is a very important one that TFH has to produce to connect with the B cells. And these are the NK cell subpopulations. These are the IELs, looking at the cytokines. So these are functional ILCs. And these are 7 different monocytes looking at, so this is the newest subset, 181, and CD16 positive and double negatives. These are M1, M2, and these are the suppressive myeloid cells, which are neutrophil based. And these are the 5 different DCs, looking at CD141, DC3, DC2, and these are the AXL, PDC. So if we don't know which one is talking to whom, and we are just, especially when we are so excited with CAR T cell therapies and all that, CAR T cells is a gamish. It's all T cells. How do you know out of these 150 markers, all those CAR T cells expressing what? Why is so much variability? I have just seen 50 patient data on BCMA CAR against multiple myeloma. A soluble BCMA has a very high level. It doesn't matter, he got a response. Should I say that is antigen-specific response or it is non-specific immune response because they dump so many cytokines? It's very hard to say. We don't even know whether they're pure population of CD4 or CD8 or what are they expressing, why will they all go off in 2 or 3 months? Why will they go up very quickly within a week or 2 in CAR T cells when they are given in 1 dose? So there are a lot of questions we don't even ask. I understand the clinical aspects of it. As long as patient get the response, we'll be happy with it. But those who don't get the response, if you don't ask these questions and don't study in detail, it's not that simple. So I wanted to caution. So there are a number of them I haven't shown, which we are just set it up. And I'm not just studying one method. We are doing multiple methods. So you could do multicolor. There are restrictions. You can only do 15 colors in a tube. You have to do compensations and set up FMOs. You can do site-off. Right now I'm doing 50 colors in one single tube. I can go up to 150 colors or metals in one single tube. So I can tell you, you give me your epitope matched or peptide tetramer pulled cells. I will tell you all these 150 markers in less than an hour. That's important to know. So that way you will understand the biology. I can also do the single cells sequencing with hashing. Not only I know these 200 markers on the surface, but also I know, few thousand genes, what they are expressing and what they are not expressing at the same token in less than 2 hours. So these are the detailed analysis. That's why we don't have like a cholesterol levels. If you pass that, you have a heart disease. If you don't have it, whereas immune system, you tell me, how do I give you my blood, tell me if it is healthy or non-healthy. It's not that simple. We can't tell that. So I'm trying to see if we can gauge that and tell you a little bit more information in an efficient way, so that way patients can get benefit of it. Okay, site-off is a good one. You don't have to compensate anything, but at the same token, it also has its own. Some of the antibodies don't conjugate with the metals that efficient. Metals are not pure, and you have to adjust some things, but it is better than flow cytometry. It's very easy to see where the drawbacks are. So I will show you, this is a healthy young one. This is a healthy old one. There are some differences. You could see it very quickly. They're not huge, but if you look at the myeloma patient, you could see this is the CD4 compartment memory cells. Look at, and almost monocyte compartment is huge. And so it tells you the age. We know that we have some idea as you get older, your T cell, B cells in the bone marrow production goes down, monocytes goes up. Is that an inherent problem? If you look at it, certain aspects of why MGUS itself is already beginning in the CD4 compartment, certain drawbacks, even though it is not showing the symptoms. And then most of these RNA-seq and single cell sequences shows those clones already present in the MGUS. How come they are not showing any symptoms? Who is holding? What is the immune system compartment are important in that regard? We need to know some of those things in order to conclude certain things. Okay? So detailed analysis, you have to come out of your box. Yeah, it's good to determine epitopes. It's good to determine peptides. I synthesize peptides to make immunocontraception in Texas. To cats and dogs, not to spade them, we could just give them a boost, one shot, and it will. We developed. Lloyd Benson Children's Company, and we patented, and GSK bought it and put it in the shelf, and then company is closed. So some of these don't come, but detailed immune study is very important. You have to know they don't sit in one room or one petri dish and work. They work with all sorts of things, and how well you are eating, what type of normal flora you have, and how much stress you have. Are you running? Are you eating properly? All these will tell you whether you will have a cancer or it can be cured. Okay, thank you very much. This is my friend who died, so thank you.
So thank you, very interesting talk. I'm thinking, so if we look at the immune system, we can characterize a lot of cells. We can say these are different than these, and therefore we have 6 different DCs. But we're looking at a snapshot. Do you have any idea? I don't. How dynamic is that situation?
We could tell, that's what I'm saying. You
But
give me any condition,
Yeah.
right? I challenge this person who came from Belgium.
Yeah.
He can tell whether a patient, before he put the joint together, whether this patient will heal or not. I told him, I'll tell you one hour before you tell him whether what type of a T-Rex he has, and those are the type of the T-Rex will help healing, and these are the type of the T-Rex won't help. So I can tell you any condition, because these technologies are there.
Yeah.
Within an hour or 2 hours, I can do 130 markers, very efficient.
Yes.
So that is important to know, not just, yeah, it's a class 1 molecule and peptide, whether gamma is produced or
Yeah.
not. Whether during that, let's take for example CAR T cells. Do I produce the CAR T cells and activate them 4 hours or 4 weeks?
Yeah.
You have no clue. There are companies, there are almost a dozen companies right now have a CAR T cell production. All have a different protocol. Nobody knows how many of those CD8 cells who are expressing combination of PD1 and TJ and LAG3 and producing a lot of IL-10
Yeah.
or even FOXP3 expression. They're giving to the patient, not knowing all that. And I asked one of the very prominent vice presidents, why don't you do your pre-product before you give the patient? It only takes an hour for me to tell you what you have, how clean it is.
But how does that situation stay the same? Like if I characterize
Right.
and I say I have a type 1 DC, does that DC always stay a type 1, or can it change? Can it grow? Can it mature?
Right. That part is still dynamic, right? We know that immune system has to be at a threshold level.
Mm-hmm.
Then only it can rise, right? That's why you come to the hospital with one infection, you go home with second infection, because during this phase, you acquire bacteria, your system will not react unless it's come down. So it has to come to the threshold in order to rise. That's why you get these infections. So it's the same thing. We know that PD1 is very important in the expression at the height of the immune response. That PD1 levels are very high at the high of the immune response. You take a hepatitis B vaccine today, you come back next week, I will show you tons of PD1 positive CD8 cells after 1 week. That is very important, so that way system will go down. So if we don't even know what they are expressing, what is consistent? My goal is, if there are 6 different DCs, to do 6 different setups. At least you could see that instead of taking that. There's only one DC, and I don't know whether he will provoke IL-10 producing CD8 cells or gamma producing, or will it continue that way or not. But at least if I show you DC4 is important for gamma production CD8, then you could take it that, and then I showed you another DC is important in producing type 1 interferons or IL-10, then we have some handle, better than before.
Yeah.
So that's all I ask for.
No.